Okay, so it's moving right through the membrane according to simple diffusion. And so we talked about some factors that would affect how fast that can occur. We spoke about the strength of the driving force, right? How big of a gradient do we have? We talked about the amount of surface area available for that transfer. And then the last factor that we left off here at was the membrane permeability. And then within the membrane permeability, we spoke about some other things that could influence that in and of itself. So things that would affect how likely something is to go through the membrane, whether it's lipid soluble or not, right? Lipid soluble meaning that it could exist within that lipid bilayer, whereas if it were polar, it cannot exist within that lipid bilayer, and so it's not going to be able to go through that membrane. We also spoke about the size and shape of particles. If they're big and bulky, they won't go through. If they're small, they can. And then the temperature, not so relevant in our bodies, but the temperature does increase the activity, the thermal energy of particles, and so they will be more likely to move in general, and then more so to move through the membrane as well. And then the last thing was the thickness of the membrane. Really kind of makes sense, right? So if something is thicker, it's going to be harder to get through. This is why most of our membranes that are adapted or suited for exchange, gas exchange, like our capillaries, like our alveoli in the lungs, they're very thin membranes to facilitate that permeability, that molecules and particles can go right through those membranes, okay? And then looking at this graph here, we're comparing the net flux, so the net movement of particles, to the size of the concentration gradient. And then we're thinking specifically about how permeability is influencing that, right? If we have a strong gradient, we're going to see a lot of movement of particles. But if the membrane is really permeable to that particle, we're going to have an increase in the rate of that movement as well. So even if we have a low permeability, we'll still see movement of particles. But if we have a high permeability, we're going to see a faster rate of that net flux moving along with the concentration gradient. Okay. So we're going to look at these checkpoint questions here. And so just kind of going over what we've just spoken about, let's review some of this with a few questions. So the first one, in simple diffusion, do individual molecules always go down their concentration gradient? And then why or why not? So what do we think as far as simple diffusion and the direction of movement of particles according to that concentration gradient?
In simple diffusion, sometimes molecules don't follow the concentration gradient because it depends on the pull that is bringing the molecules in.
So when we're talking about simple diffusion, this is going to always be down the concentration gradient. So let me just clarify that. What you may be talking about, I think is what I'm picking up from your response, is that you can have particles moving to either side, which is true. We spoke about that. You can have one-way flux. But as far as net flux, particles are always going to move down their concentration gradient, okay?
Okay.
Yeah, sure, just to clarify that there. And then the reason is because that's going to be the spontaneous movement, right? That's going to be the natural flow according to said gradient. Although you may see one particle bouncing from one side to the other, that one-way flux, the net accumulation will be according to that gradient. Question 2, which properties of transported molecules affect membrane permeability in simple diffusion? So some of the properties that we kind of just went over. So what are some things that would make it more or less likely for a particle to go through the membrane, for the membrane to be permeable to said particle?
Thickness of membrane.
Yep, so I'm hearing thickness of the membrane, that's perfectly correct, size and shape. So as far as the first part of the question, what properties of the particle, so that would be size and shape, that's correct. And then as far as the second part of the question, what properties of the membrane, that would be thickness of the membrane, okay? So size and shape of the particle itself, the lipophilic or hydrophobic nature of the particle itself, And then in terms of the membrane, it would be the thickness of the membrane, the amount of surface area exposed. Okay. Yep, so someone also is mentioning here in the chat, lipid solubility, size and shape. Excellent. So I hope that question was clear. We're talking about two different factors here. So factors of the molecule or particle, and then factors of the membrane itself. Okay. Number 3, for a substance crossing a tissue by simple diffusion, which of the following, choose all that apply, cause the permeability to increase. So we've got increased tissue thickness, decreased size of diffusing molecules, decreased lipid solubility of diffusing molecules, and then increased temperature. So which of these would cause the permeability to increase?
Temperature?
Excellent. Yes, so an increase in temperature will definitely increase the thermal energy of those particles. That will increase the permeability.
Decreased size.
Perfect. Decreased size of diffusing molecules. So if we have a smaller molecule, it's going to be more suited, more adapted to go through that membrane. That would too increase the permeability. And then the other two responses, increased tissue thickness, that would go against permeability, right? That's going to be more stuff in the way. A thicker membrane is going to actually decrease permeability. And then the final option here, decreased lipid solubility. If we decrease the lipid solubility of that molecule, again, it's going to be harder to go through the membrane. So it's not going to be able to exist within that lipid bilayer. And so that would actually decrease permeability. Okay, so the two correct responses as far as increasing permeability here would have been decreasing size of diffusing molecules and then increasing the temperature overall. All right. So we're going to move on to objective number 3 here. We're comparing the movement of molecules across the plasma membrane by carriers and through channels. Now, I want to reiterate before we start here that this is still simple diffusion. This is still passive movement. This is still movement along or according to or down the concentration gradient, although we're needing some help, although we need some structures in the membrane to facilitate this type of diffusion, okay? So simple diffusion, facilitated diffusion, and then channel diffusion, these are all mechanisms of passive transport. Really important to make that distinction. So let's start talking about facilitated diffusion here. This is movement through a carrier. Again, it's passive or down the concentration gradient. But because some of these particles are bulkier, glucose is a really good example, they can't go through the membrane, and so they need some help. And so we have what's called a carrier or a transporter in the membrane. It's actually an example of one of our transmembrane proteins. You remember those proteins that span the membrane, span the width of that bilipid, that phospholipid bilayer. And so these are embedded within the membrane, and they facilitate the movement of some of our bulkier molecules like glucose. So it has a specific binding site for the particle on one end, and it can bind to one side. It'll have a conformational or configurational change, and then it will kind of spit the molecule out on the other side of the membrane. So here's that example. We've got glucose here, a glucose transporter or glucose carrier embedded within the membrane, right, spanning that both sides of that lipid bilayer there. And so glucose will bind to one face of this carrier. And this carrier is really a protein, so just make that note as well. It's just a protein that is suited to do this. It'll bind to one side of this carrier. It'll have a shape shift or conformational or configurational change, and it will open the other side of that carrier into the cell, and that will kind of spit that glucose onto the inside of the cell. And so this is an example of facilitated diffusion, right? It's moving down its concentration gradient, but it's needing some help to do so. And that's where these carriers or transporters come into play. Now, when we look at the factors that affect the rate of facilitated diffusion, that is going to be dependent upon the rate of transport of each individual carrier. So how fast can each of these carriers get glucose across? And so it's going to also be determined by the number of carriers that are in the membrane, right? And so as we can see here, the graph is going to look a little bit different than our simple diffusion graph did. So just kind of thinking back to our graph for simple diffusion, actually, let's kind of jump back and just remind ourselves here, the graph for simple diffusion is a completely linear curve. It is directly proportional. So as big as the concentration gradient will get, that's as fast as particles will move. That's the amount of net flux that we're going to see as far as that movement. But when we look at the graph here for facilitated diffusion, this curve eventually flattens out. And what that tells us is that the concentration gradient can only get so large, and then at some point we're going to have a max, this maxing out of our transporters. We're going to have the maximum number of our transporters. They're going to be moving as fast as they can, and then that's where we're going to see that curve flattening off, such that if the concentration gradient gets bigger, we will not see an increase in the weight, right? There's going to be a limit to how fast that transport can take place, okay, because of that limiting factor, which is the weight of each individual carrier and then the number of carriers that can actually, physically be in the membrane at any given time, okay? And then once we max that out, that curve will flatten off. A really good example of a facilitated carrier is the glucose transporter 4, or the GLUT4 transporter. This is a transporter made in the rough endoplasmic reticulum. And if you remember from prior knowledge, the rough ER is where we make proteins or molecules that are going to be embedded into the membrane, okay? And so this is where these GLUT4 molecules are synthesized. They are packaged into vesicles. They are exported and embedded into the membrane to facilitate this glucose uptake inside or into the cell. And so this is what the action of insulin stimulates. So insulin actually tells the cell to insert more of these GLUT4 transporters into its membrane so that glucose can be uptaken and used for cellular respiration, to make ATP, et cetera, okay? And so this is one of the reasons why we have in diabetes mellitus or type 2 diabetes, there's a lack of insulin, meaning there's a blunting of that signal. And so we have less of these GLUT4 transporters inserted into the membrane. This translates to less glucose being uptaken and used appropriately to make ATP. And so instead, we see more of that glucose floating around in the bloodstream. And this is where we get the high blood sugar readings and the high blood sugar levels from, okay? That glucose can't get into the cell because there's no insulin sending the signal to make the transporters to uptake that glucose appropriately. Okay, and aside from just having glucose in the blood in terms of those high levels, it becomes more detrimental because glucose can be damaging to our blood vessels. It can be damaging to the small vessels in the retina of the eye, in the kidneys. And so it's not ideal to have glucose floating around in our blood, in large quantities indefinitely. It's really supposed to be uptaken into the cells and then used for ATP production. All right, so that was our transporters, and that was basically facilitated diffusion or facilitated carriers. The second mechanism here is going to be diffusion through channels. And so it's a similar mechanism in that these particles need some help getting through the membrane, but this is a different structure altogether. So this is also passive, right? So keep making that note. This is also passive, moving down the concentration gradient. The characteristics of the channel are that it's also a transmembrane protein, so another type of protein embedded into the membrane as well. It functions like a passageway or a pore or a channel, as the name indicates. And so it's going to be substance specific, and it's going to allow different types of molecules and particles to enter to the cell. Okay, and so looking at this image here, we can see the cell membrane, that lipid bilayer. We can see this channel or pore. And as we can see, it's a different configuration than the carriers were altogether. It's going to be more of a passageway or a pore type configuration. There are different types of channels. We've got aquaporin channels, and these are channels specifically suited for water uptake. We'll talk about aquaporins when we look at the kidneys and how water is reabsorbed into the kidneys via these types of channels. There are also ion channels, so channels that specifically allow the movement of charged particles such as sodium, potassium, calcium, chloride, etc. And then within ion channels, there are further subdivisions. We've got leak channels, we've got gated channels that have a specific mechanism to unlock them. There are different types of gated channels that we'll get into later on. There could be voltage gated, there could be ligand gated. And then the third type here are bidirectional channels. And then these are just channels that facilitate movement in either direction, right? So in or out of the cell. And then getting into the factors that affect the rate of transport via ion channels, they're going to be similar to what we saw for those carriers. It's going to be dependent upon the specific rate of each individual channel. So how fast can that channel transport these particles, whether it's water or an ion, through the membrane? It's also going to be affected by the number of channels in the membrane. Again, how many of these channels can physically fit in the membrane to facilitate that movement. And then it's also going to be influenced by the state of that channel, whether it's opened or closed. And then that is going to be more relevant as we speak about some of these gated channels, right? So we can have specific mechanisms or specific conditions that will dictate whether a channel is open based upon its gating mechanism, okay? All right. So I'll pause here a little bit and see what questions we have, and then we'll look at some checkpoint questions. All right, so the first checkpoint question here, what is the difference between a carrier and a channel? And then how are they similar?
A carrier and a channel, they both carry particles, but a carrier specifically carries particles, I would say. I'm not exactly sure.
That's okay, you're on the correct lines there. So a carrier is going to pretty much, and then if you look at the name of the word here, this will kind of help with that distinction, but a carrier is going to be that facilitated diffusion. In other words, it's going to help that molecule get through. It's going to do something in and of itself. It's going to shape shift in order to facilitate or help or guide that particle through. So you were perfectly correct in the beginning there in terms of that, that train of thought. A channel, on the other hand, is kind of just standing there, right? It's just a passageway. It's just a pore. It's not going to do anything to help the molecules get through. It's just going to exist, if you will, right? And so that's kind of the fundamental difference between these two. And then ways that they're similar is pretty much what they do, right? They're helping things get through the membrane. We can think of the carrier playing more of an active role in that process, and then the channel playing more of a passive or laid back role. It's just kind of existing there and allowing that molecule to get through. Okay. Other ways that they're similar is that they both require that passive movement, so along the concentration gradient. And then they are also going to be, the factors that influence the rate will be the same, right? The number of carriers or channels, the individual weight of each of those carriers and channels, okay? The second question here, as the concentration of a substance increases, the flux of molecules transported by carriers increases at first, but eventually, this should be but, excuse me, not by, increases at first, but eventually levels off. And then the question is, why do we see this? Why is this? So thinking back to those two curves that we compared, those two graphs we compared, when we see movement of these substances via carriers, we see a linear curve at first, but then we see that flattening off of the curve. So what is that flattening off due to?
It's a limiting factor.
Excellent. Yes, it's going to be a limiting factor, right? Remember that we only have so many carriers. And then when those get maxed out, or when they're, transporting as many molecules, let's say of glucose as they can per minute, we're then eventually going to have a flattening off of the curve, such that even if our concentration gradient starts to get bigger, we cannot physically move any more molecules any faster because these transporters are maxed out, okay? Number 3, when an uncharged substance such as glucose enters a cell by facilitated diffusion, does it go up or down its concentration gradient? So if we've got a movement of a molecule like glucose by a facilitated diffusion, is that moving up or down its concentration gradient? So this should be down its concentration gradient, right? Always, always, always down its concentration gradient. So I'm gonna just kind of repeat this here again. When we move particles, whether it's simple diffusion, going right through the membrane, or facilitated diffusion, requiring some help, or through a channel, which is another way of acquiring some help, all those are examples of passive movement. So the particles themselves are still moving according to their concentration gradient. So from high concentration on one side to low concentration on the other side. The only difference is that for facilitated diffusion and for channel transport through channels, that is going to be where that particle or molecule needs some help getting through the membrane, but we still have that movement according to that high to low concentration. Okay, which is what really makes it passive. That's the part of this that is that passive transport.